Hello everyone, in this video I will discuss how helper T cell and cytotoxic T cell get activated and go through further processes. Uh, in the previous video I discussed about the effector T cell and memory T cell. In the effector T cell there is basically a helper T cell and cytotoxic T cell. So now first discuss about the helper T cell. Once this helper T cell is get activated and in the beginning basically this helper T cell, let's say this is a helper T cell, and this helper T cell is activated by this MSC class 2 molecule, and this MSC class 2 molecule is basically present a antigen. This antigen is basically exogenous antigen, and MSC class 2 molecule and antigen presenting cell like macrophages or B cell always present or always contain MSC class 2 molecule. So this MSC class 2 molecule here basically present the antigen. Here basically this is foreign antigen, and this foreign antigen is basically recognized by helper T cell. Then in the surface of the T cell there is a receptor called T cell receptor. This T cell receptor is here bind with this antigen, or we can say that here bind with this exogenous antigen. Once the binding happen, then it will go through the clonal selection process. In the clonal selection process, activated helper T cell this activated helper T cell is basically converted into memory T cell, and the other part of this helper T cell will converted into effector T cell. Now, this effector T cell is then basically, or effector T cell, or we can say that this is basically activated helper T cell. So, this activated helper T cell then secrete cytokines. So, here these red dots or orange dot or green dot, these are basically cytokines. So, let me write this. So, these are basically cytokines. So this effector T cell or activated helper T cell then secrete cytokines, and these cytokines or this chemical further activate neutrophils or macrophages or cytotoxic T cell or B cell. And in this memory T cell is basically happen in both process. In activated helper T cell or activated cytotoxic T cell, both produce the memory T cell. These memory T cells are basically long-lived cells that remain in the body after the initial infection has been cleared. They basically remember the specific antigen. Suppose this is antigen, and again let's say in after few years or few months, this same antigen inserted into our body or get infected, our body get infected by this antigen. Then this helper T cell basically remember this antigen after first infection. So as this memory T cell already recognize this antigen, they remember and this specific antigen and can respond more quickly and effectively if the same pathogen is encountered again. So here we basically see that how this helper T cell get activated and then further go these processes. So basically how this help in the killing of the virus or bacteria the or the protein or foreign antigens. So these neutrophils or macrophages basically get activated by cytokines. Then so these neutrophils and macrophages go through phagocytosis and engulf the bacteria or engulf the antigen. Then this killer T cell, or we can say that a cytotoxic T cell or cytotoxic T lymphocyte, this cell basically directly kill the pathogen. And B cell here we see that here B cell produce after get activated by cytokines. B cell produce antibodies. These antibodies then bind with the antigen or bind with the virus or bacteria, and it block this. And antigen basically do what? Antigen basically block the surface protein of the bacteria or virus, so then the bacteria or virus cannot go through the or cannot bind with the receptor of the cell of the normal cell. Okay, so this is basically the activation of helper T cell and its action. Now let's see the cytotoxic T cell activation and its action. So this, wait a minute. So here this is cytotoxic T cell. This cytotoxic T cell also contain T cell receptor, and this cytotoxic T cell only recognize the antigen from MSC class 1 molecule. This is basically restricted, or this is a constant thing. This can be changed in all types of organism. We will see this type of interaction that always cytotoxic T cell recognize antigen or recognize foreign antigen from the MSC class 1 molecule, and this MSC class 1 molecule is contained by all around all types of nucleated cells. So here we see that MSC class 1 molecule contain a foreign antigen. This foreign antigen is then binds with the T cell receptor. Then the cytotoxic T cell will get activated as it recognizes the antigen which is presented by MSC class 1 molecule. Then this cytotoxic T cell will proliferate and differentiate into cytotoxic T lymphocyte. This thing we also call that cytotoxic T lymphocyte. This cells also called cytotoxic T lymphocyte, or in short from CTL. Okay, and here this will go through clonal selection. In this clonal selection process, there will produce activated cytotoxic T cell and memory T cells. This memory T cell will act like before we see in the B cell activation or proliferation. So this memory T cell also work similar like that, and this activated cytotoxic T cell now directly attack the infected cell or cancer cell, and this is we can say that this is basically lethal hit. And this cytotoxic T lymphocyte basically induce apoptosis. So let me write it. Okay, this, okay. So here after recognize the target, the activated cytotoxic T cell or cytotoxic T lymphocyte then induce apoptosis. So here let's see that first this target recognition, then second this cytotoxic T lymphocyte use to main pathway to induce apoptosis. This is in target cell. So here it basically, this two pathway is basically perforin and granzyme pathway. First one is perforin granzyme pathway, and the second one is phosphorus ligand pathway. So in this perforin and granzyme pathway, perforin basically make a pore in the target cell's membrane. So this perforin, this chemical, suppose this is perforin, this perforin basically make a short pore or make a pore in the surface of the infected cell or self-altered cell, and granzyme uses this pore and activate. Granzyme basically use this pore and activate caspase caspases, and then this caspase initiate the apoptosis pathway. And in phosphorus ligand pathway, this basically there is basically a fast ligand and fast receptor. There is a, let me describe this. In the first phosphorus ligand pathway, the target cell, basically this is target cell. This target cell contain fast receptor, and cytotoxic T lymphocyte contain fast ligand. So once this fast ligand binds with the fast receptor, then it will proliferate into, or it will lead to activation of caspases and apoptosis. So that's all. And so this is basically the whole process of cell-mediated immunity, from recognizing a foreign antigen or recognizing a self-altered cell and kill them and make our body free from foreign antigens, or let's say virus or harmful virus or bacteria, or protect us from cancerous cells. And that's all I